Okay, folks, 7 o'clock here, and so we're going to go ahead and get started as people are still logging in and entering their info into the chat box there. Thanks for joining us. Tonight's Farminar is, as always, brought to you by Practical Farmers of Iowa. We put these on every Tuesday night during the fall and winter months. I'm Steve Carlson. I work with PFI as a beginning farmer and program associate. And I'm going to speak pretty quick here about Practical Farmers of Iowa before I turn it over to Paul Ackley, who Paul's a longtime PFI member that farms near Bedford, Iowa, down in Taylor County. And Paul's going to talk for a little while about his operation and how he has used the Haney Tests to inform some of the decisions he makes on his farm. And then we're going to pass the mic to Sarah Hargreaves up in Canada. Sarah is a soil microbial ecologist who focuses on agricultural ecosystems. So she'll give us a kind of a crash course on soil microbiology and then talk about some of Paul's test results and how to interpret them. So like I said, we hold these every Tuesday night in the fall and winter months at 7 p.m. Central Time. And as you can see, we've just got a couple left for this fall series. And then we're going to take a few weeks off and then start back up in January. So if you want to see full descriptions of these two Farminars coming up and the speaker bios, then you can check the Farminar webpage that you used to log in here. And then look for an announcement coming from our newsletters or our website or social media or wherever about the next round of speakers and topics that'll start mid-January. So if you're not familiar, Practical Farmers of Iowa started back in 1985, so this is our 30th anniversary. And PFI is a nonprofit organization made up of farmers and friends of farmers. Our farmers come from farms of all sizes and enterprises, livestock, row crops, horticulture, everything in between. And our farmers and friends of farmers are from all across Iowa and across the Midwest and throughout the United States, really. So as a nonprofit organization, our mission is strengthening farms and communities through farmer-led investigation and information sharing. And we use this focus to help farmers practice an agriculture that benefits both the land and the people. Our values at PFI, welcoming everyone, creativity, collaboration, and community, viable farms now and for future generations, and stewardship and ecology. And so if you like the kinds of things we do at PFI, you should become a member. Your membership supports our organization and allows you to tap into our network of members, which now has grown to around 3,000 people. And so you'll get, as a member, our newsletters, discounts to our events. We do just dozens and dozens of events throughout the year, and the opportunity to participate in our research projects and all of our programming. Our members really do help guide our programming, which these Farminars are included. So these topics are taken from our member feedback, just along with pretty much about everything else we do. And so don't forget to check our event calendar on our website there, practicalfarmers.org/events, for both PFI and non-PFI events. It's chock full of resources and things that are happening across Iowa and across the Midwest. So if you've got free time on your hands nowadays, go and check that out. And then, of course, the main event that we like to talk about this time of year is our annual conference, which this year the theme for that is celebrating 30 years of farmers teaching farmers. And we've got a few dozen sessions planned. Many of them, if you're interested in this topic tonight, you might like for instance, Paul Ackley, one of our presenters tonight, is going to be on a panel with other Iowa farmers talking about cover crops for Iowa. And we have a few other soil health sessions. So you can get all the info about this conference coming up on our website right there. And you can get registered and all of that on the website. So check that out. And before we get started here, the basic rules. Thank you, everybody, for entering your location and your email into the chat box. Like I said, we use that for grant reporting purposes, and then we also help evaluate our Farminars and get feedback for what you want to hear about next season. So if you have questions tonight for our presenters, go ahead and enter them into the chat box. You can type your questions in there at any time. We are going to reserve the final 30 minutes to go through questions. So if our speakers happen to see your question and want to address it while we're going, they might do that. Otherwise, we'll try and get back through all those questions at the end. So type them in or save them. And now I'm going to pull up Paul Ackley's presentation and let Paul take it away.
Am I on, Steve? Okay.
Yeah, yep, you sure are, Paul.
Okay. Thanks for asking me. We'll share some experiences we've had in this process. We're not experts or anything like that, but we kind of been working at it. We farm in the southwest corner of the state, just a few miles from Missouri. The farm is about 640 acres owned and about 400 rented. I would say all of our land is probably classified highly erodible, and it would probably, if you were to look a little further and deeper, you would say it was degraded, as most of the land that's been farmed for the last 100 years, I think, would be class degraded. Became aware of that, I guess, when we went through some, included some fence rows in a field and brought some pasture in that had never been plowed. One thing that I guess we've been working on the last few years is building soil health, or trying to build up our soil. And I heard Dwayne Beck say one thing that was always stuck with me, and it kind of answered a riddle for my father, who said this country was too good to ranch and too poor to farm. Dwayne Beck's theory is, don't change your land to fit your management, change your management to fit your land. And so that's kind of what we've been trying to do. We haven't built a lot of terraces, especially in the last 20 years, and there are quite a few terraces in this area. We've been doing it with cover crops and lengthening our rotation, planting some alfalfa and a mixed grass. We have about 80 cows we calve in May and June. Typically the steers would be sold the next March as feeders. We've been keeping all the heifers and then selling the ones that fail to breed. We have about 500 acres of out of that ground we have about 500 acres in diversified crop rotation of corn, soybeans, and winter wheat. And soybeans typically make up half of those crop acres because we split the other half between the corn and the winter wheat. The last 2 years we've aerial seeded just about everything. That way we don't have to go over anything with the drill after harvest. We got a really good stand this year. We use about 50 pounds of cereal rye, add a pound of purple top turnips, and a pound of rape. There are some areas that tend to fill in later, that it doesn't all come up just perfect, but I think it's better than going over some of it with the drill. The cows really turn up their nose at a stalk field if you've been over it with a no-till drill, so that's one reason. As long as we can get a decent stand, we'll stick with the airplane, I guess. I credit the CSP program from NRCS partly for getting us started with adding a small grain. If you read between the lines on that CSP program on some of those practices, there's encouragement to start diversifying and doing something besides corn and soybeans. And that was the first thing that gave us a incentive to go ahead and try some winter wheat. And we followed that first wheat crop with a very diverse rotation of or diverse cover crop with a lot of broadleaves, really too many broadleaves, because I wanted to follow it with corn. We liked the results, but we got a lot of grazing out of that thing. We Typically we'll plant that mix the middle of July. This year we didn't get it seeded until the middle of August, and we're not going to get a lot of tonnage out of it this year. The background picture for this slide would be that cover crop mix that was seeded about the middle of August into wheat stubble. Wheat stubble was visible there. Oh, it got about knee high. Of course, the forage sorghum winter killed about the first frost, and everything else has stayed green until just the last few days. We have been able to drop the phosphate and potash following winter wheat, whether we go to soybeans or corn. That's kind of a flip of the coin, or what we think the field needs. That was one of the things that helped make adding the wheat to the rotation economically work. We've also, I thought we would get to drop one herbicide treatment, but we have not got to do that yet, but I think we will in the future get to the point where we'll have the weeds under control enough that we can drop out one herbicide application. We were putting on about 50 to 60 pounds of phosphate and about 30 of potash. That would have been our normal maintenance program for those two elements. The diverse cover crop mix usually includes some purple top turnips, forage collard, probably cowpeas is one I like, and maybe another nitrogen fixer or two, and then some taller forage sorghum for grazing. The forage sorghum has a real dense root system like a grass, and of course I don't use any radishes. I'm not a big fan of radishes. If you put a lot of broadleaves and a lot of brassicas in it, this wheat stubble will be gone by next spring. If we go more with some of the grass, there will be some wheat stubble there next spring. It varies with how we tilt that mix. We also add sunflowers for the taproot. I really like those. Cows, if they get to go to seed, that'll be one of the first things the cows will pick is the head in the winter. We like to start grazing about the 1st of November if we can. We've had to shut them out of it this year. We've gotten so much rain lately. We've got March 1st marked on the calendar. Cows come out then or shortly after or before if the frost goes out of the ground. We don't leave them there and try and straighten things up with a disc or field cultivator or something. We tried that once. We had some cows grazing rye until the middle of May, and I still went ahead and no-tilled it, and we had a really poor soybean crop after that. So we just made that one of the rules that they come out. Usually if the frost leaves the ground by the 1st of March and is somewhat pugged or tramped up, we'll usually get another light freeze, and if the cattle are gone, well, that'll take care of some of that compaction or top hardness that we have. Last summer we seeded for the first time to pasture that year into soybean stubble, 2014 soybean stubble. We seeded oats, alfalfa, red clover, alsike, clover, brome, orchard grass, and timothy, and used it for grazing. It was way too heavy on the legumes, but we used some yearling heifers that we were saving. We got about 200 pounds of beef per acre. I think that's going to be another alternative to a small grain. Now, strangely enough, that soybean stubble had rye flowing on it last year, and it had a fairly decent stand. I, at one time, didn't think that was possible to do that, but it worked out really well. The rye got up and got going and got ripe. The calves didn't like it at all, so they just tramped it down and ate the oats and grazed the green underneath. So we got added some biomass and some carbon to the soil by just trampling that mature rye down. In about 2012 or 13, we saw Rick Haney at No Till on the Plains and listened to him, and we'd been cutting our fertilizer probably a little anyway, or it would always been on the low side of the recommendations, especially for phosphate and potash. I think I got really too low on the nitrogen way too fast, and we hurt ourselves sometimes on corn yields. But anyway, it makes sense. Rick's, as he explains it, Mother Nature doesn't use an acid to strip the nutrients out of the soil. It uses water, and that's what he uses, and he's trying to mimic nature in her own surroundings. We feel like that when you start getting more organic matter in the soil, the organic matter starts going up a little bit with the cover crops. Definitely makes a big jump after a diverse cover crop and grazing, that probably the standard soil test is probably not catching all of it. And I do like the Haney test. It shows you exactly what the difference would be between the nutrients available, what it shows, and what would be available or on a soil test, standard soil test. But I heard Dr. Ray Ward, he had done a correlation study between the Haney test and the standard test, and he looked, you could see the line. It was very, very close. There was definite relationship. He said, I don't think we've done too bad of a job with what we had to work with at the time. I think the goal for using the Haney test was to see what we could do. I also heard Dr. Christine Nichols, who was at that time with the Ag Research Service at Mandan, North Dakota. She was going into all the microbial life in the soil, and I was following her, and I thought, good grief, she is going to tell us we don't need to use fertilizer. And she said, yeah, if you keep applying commercial fertilizer, the microbes just stay lazy and don't do what they're designed to do in the soil. And so that was another reason, I guess, to use the Haney test. We have been grid sampling our soils since 2000. Each field was typically grid sampled every 4 years in 2.5 acre grids, and I was only applying maintenance levels of phosphate and potash, and we'll flip to this next one. And there's kind of an average of one farm. Some of them are really low in phosphorus. The first 4 or 5 years was the big jump, and then it got slower. These would have had a cover crop on between corn and soybeans. There would have been no wheat in this rotation. We didn't start, I think we planted the first wheat crop in 2009 in that fall. But they kind of went along those lines, and so I guess that's about where I'm at. So Steve, I think we're ready for Sarah, unless we've got a question.
Yeah, thank you, Paul. I am pulling up Sarah's slideshow right now. And if you do have questions, Sarah's going to use Paul's tests as an example for you. But if you do have questions for Paul, keep thinking of him, and we'll come back and do some Q&A after Sarah talks about microbial activity and uses some of Paul's info as an example here.
Oh, hi, everyone. Can you hear me? Hello? Yeah, okay.
Yes, we can definitely hear you. Thank you. Yep. Sorry,
great.
we got
So I'm really thrilled to be talking to PFI again. I lived in Iowa for 10 years, and it was the PFI members that really inspired me and my husband to start farming. We are just happen to be located in Ontario, Canada now, which is where I'm originally from. So it's great to be back in the PFI land, even virtually. Stefan and Steve asked me to present today and dig a little deeper with you all on the microbial ecology of the Haney Test, and specifically to address this equation of how microbial activity translates into nutrient turnover in the soil and ultimately nutrient availability for plants. And so I'm going to briefly just talk about what I mean when I say microbe. I think it's important we're all on the same page about what a microbe is, and then expand more on this equation of microbial activity equaling nutrient turnover, explain what specifically drives crop available nutrients in this activity turnover relationship, and then how the Haney Test specifically measures microbial activity and the aspects of microbial life that it captures. And I'll use Paul's results as an example. And then I'm also going to touch on the phospholipid fatty acid test, which is a measure of microbial communities, and talk a little bit how it complements the Haney test, and also use Paul's results. Paul's done a really amazing job at sampling and taking advantage of the Haney test and the phospholipid test to help him on his farm. So as I said, we're going to start off I just want everyone to understand what a microbe is. Microorganisms encompass all three domains of life. All bacteria and all archaea are microbes. And then there's one kingdom in the eukaryotes, the fungi, and they're also microbes. And over the last couple of decades, and the last decade specifically, there have been some really great technologies, and they've allowed us to really delve into the diversity of microbes, but we still haven't even really touched the surface. But what we do know is that they comprise the large majority of biodiversity on Earth, and they're really important powerhouses in nutrient cycling and nutrient transformations everywhere on Earth. Specifically in the soil, there are a couple of really important groups of microbes, and these are the microbes that I'm talking about when I say soil microbes. So there are saprotrophic fungi, or sap, and these are, and then there are heterotrophic bacteria, and there are arbuscular mycorrhizal fungi. And so this is going to start to become a pretty wordy slide. It's the craziest slide I have, so don't freak out. This isn't, my slides aren't going to be this dense, but I think this information is important. So saprotrophic fungi are decomposers, and they're really the powerhouses of nutrient turnover. And they're heterotrophic, and all that means is that they use organic material as their carbon source. This is similar to us. We eat food that has carbon, and that's what we use to fuel our bodies. They also excrete these extracellular digestive enzymes into the soil, and it's these soils that digest organic matter and lead to nutrient turnover. And then saprotrophic fungi have also been found to be really important for nitrogen and water retention in soils. And this has to do with the fact that they're slow growing, but it also has to do with the fact that they just, they're quite massive when you take into account all their mycelium and the networks that they actually create underground. They're a huge biomass of nitrogen storage, of a nice buffering of nitrogen in the soil. Heterotrophic bacteria are the same as saps. They're decomposers. They excrete those same enzymes. And some specific examples of heterotrophic bacteria that come up in the phospholipid test and are just important when thinking about farming are rhizobia. These are these nitrogen fixers that form root symbioses with legumes. And then there are also actinomycetes, and these are important bacteria because they're fungus-like. They have these filaments and create networks or soil bridges in the soil. And I just want to clarify that not all bacteria are heterotrophic, but when I'm talking about bacteria and soil bacteria, I'm really talking about these decomposers. And then I'm not going to talk much about arbuscular mycorrhizal fungi because they are actually not specifically involved in nutrient turnover, the topic of today, but they have a place in any presentation talking about plant-soil relationships, because they're such important symbionts. And I think it's important to mention them because they have a really important role for aggregation, which just does have a role in creating a nice soil environment for microbes to live in and be able to cycle nutrients. So, but as I said, when I say microbes, I'm talking about these saprotrophic fungi and heterotrophic bacteria, and I hope that wasn't too confusing. But let's get back to the really why we're here, and this relationship between microbial activity and nutrient turnover, and how the Haney Test ultimately fits in to this relationship. And so when we talk about microbial activity, I'm just going to start to break apart these pieces. Is one aspect of microbial activity is mineralization. And what mineralization essentially is microbial digestion. And so what happens is we have plant roots. They excrete these root sugars, which is a liquid carbon and a liquid nitrogen. Think ice cream, a really nice liquid source of fast energy. And this gives microbes energy to produce digestive enzymes. And just like the enzymes in our guts that break down food and give us nutrients, these microbial enzymes digest things like crop residues and dead roots. And as a result of this microbial activity, this microbial digestion, you get nutrient turnover, which is essentially just the liberation of nitrogen and phosphorus in the soil. And then you also get this decomposed organic material incorporated into the soil as soil organic matter. So nitrogen and phosphorus are available for plants, leading to crop growth. And we also see that this newly decomposed organic matter is further broken down and decomposed, and further broken down and decomposed, et cetera, until it forms humus, or a stable soil, and is incorporated into aggregates. And this is where we get like carbon storage and ultimately the beautiful texture of a healthy soil. And the second component of microbial activity is respiration, or microbial breathing. So just like us, these heterotrophic microbes use oxygen to respire, and they produce carbon dioxide. And the really beautiful thing about this part of the activity is carbon dioxide is really easy to measure in the lab. And so this is what Dr. Haney took advantage of, because we can measure carbon dioxide and we can use it as a proxy or as an indicator for how much microbial activity is happening in the soil. It's certainly not a perfect indicator of microbial activity, but it's a really good one, and it's a really easy one. And we'll go over that a bit more in a few more slides. And so just so we're all sure, all of this complete equation from liquid nitrogen down to humus is what we mean when we say decomposition. So it's this entire process of mineralization, microbial digestion, and respiration, or microbial breathing. And that's what we say when we mean decompose and decomposition. But let's step back for a second and really think about what's motivating probably most of us for being here, and what motivated Paul for looking at Haney Test. And it's this pool of nitrogen and phosphorus in the soil. So this pool of available nutrients that crops can take up that can lower our fertilizer inputs, and all these really important things. And let's think about how we can increase this available nitrogen and phosphorus in the context of this microbial activity equaling nutrient turnover equation that we have. And so to do this, we really need to think about how and what we feed the microbes. So I like to call it farming to feed microbes. And the next few slides, bear with me, folks, it's like you're going to be getting a PhD in biogeochemistry in approximately, a couple of minutes. But it's actually really not that difficult if you just think about feeding microbes like we think about feeding our livestock. And I think it's a pretty common, there's a familiar concept of feed value when you think especially of cattle. And so here's a photo of our little heifer calf, and we've just have our first 3 cows, and it's pretty exciting. But putting up hay, I had to think a lot about the quality of that hay. And we know that when you have good pasture or good hay, you require less of it to feed the same number of cows. So you have a better feed quality. And when you have poor quality pasture or hay, there's just less feed value and you can feed fewer cattle per unit. And so we're just going to like apply this simple concept of feed value to microbes. But when we think about microbes and feed value, what we need to think about is carbon to nitrogen ratios in the soil and in the organic matter. All this means is the amount of carbon there is per unit nitrogen. So a carbon to nitrogen ratio of 20 has 20 carbons in a piece of organic matter to one nitrogen. And the only tricky part about carbon to nitrogen ratios is that, in terms of feed value, is as the carbon to nitrogen ratio increases, the feed value actually goes down. And that's because microbes are wanting nitrogen in soil. Everything wants nitrogen. And so the more nitrogen you have per unit carbon, the higher the feed value. So that's sort of the only place where I think this concept might become confusing, is it's that feed value actually is a lower carbon to nitrogen ratio. And so when we look at this sliding scale of carbon to nitrogen ratios, you'll notice that approximately in the middle of the scale that I have, and it goes off, into 100 carbon to nitrogen ratios of 100 or 200 to 1, but at around 24 to 1 is where we have the sweet spot for microbial growth. And I'll just quickly explain why. So microbial cells are approximately 8 carbon to 1 nitrogen. And it takes approximately an extra, well, not approximately, it takes exactly an extra 16 carbon of respiration to maintain that cell. So 8 plus 16 is 24, and 1 nitrogen. And that's really, if a microbe has a piece of organic material that has 24 units carbon and 1 unit nitrogen in it, then it's set. It's very efficient to break that down. It can use everything in that piece of organic matter. Whereas as you get higher, as you have carbon to nitrogen ratios that are higher, microbes have more carbon than nitrogen. So any nitrogen that they liberate, that they mineralize, they're going to use it all, and there's not going to be much left for plants. Whereas as organic matter decomposes, the microbes are taking carbon out of, are respiring carbon and decomposing that carbon, and the nitrogen value actually, the carbon to nitrogen ratio actually goes down. And what happens is you pass that sweet spot for microbial growth, the microbes still continue to decompose that organic matter, and you enter in this sweet spot for plants, which is around between 10 and 20 carbon to 1 nitrogen. And this is where microbes have more nitrogen than they have carbon. And so there's just this excess of nitrogen in the soil. And it's as simple as thinking with high carbon to nitrogen ratios, it might be like you're in your chicken yard and there are 100 chickens, and Farmer Drake comes along and throws in five pieces of popcorn. Well, you're going to be hard pressed to get that piece of popcorn from 100 chickens. That's when you have a high carbon to nitrogen ratio. Whereas when you're in that sweet spot for plants, if Farmer Drake comes along and throws in 200 pieces of popcorn, it's more likely that you're going to actually get a piece of that popcorn. There's more nitrogen floating around in the soil for plants to get. And so that's how we can think about how we can feed our microbes in the soil, and what they're doing in the soil, when and how it benefits plants. Let's see what I have. The one thing that's very different than the cattle feed analogy is that organic matter is constantly decomposing. It's constantly changing. Every time a microbe releases an enzyme and that enzyme acts on a piece of organic matter, the carbon to nitrogen ratio changes. And so what you do is you have this sort of continuum of levels of decomposition in the soil. And so the key really to keeping organic matter available in that sweet spot for plants is to have this constant supply of living plants and roots year round. So I just have this arrow moving forward to just show that when you have a diversity of living plants and roots, and you have cover crops in winter or perennial plants planted, and you have this influx of fresh organic matter into the soil, microbes can then continuously decompose organic matter from these higher carbon to nitrogen ratios down through their sweet spot and then through this sweet spot for plants. So you have a constant supply of organic matter that's being that has extra nitrogen, and the plants can actually therefore have a nice supply of inorganic nitrogen and phosphorus. And so just putting it in the context of this equation we were looking at before is when you have this diversity of living roots and plants, these roots are then able to are excreting this liquid nitrogen, these sugars. This gives microbes energy to make enzymes. It also produces this fresh organic material in residues and dead roots. This makes microbes really happy and active. They mineralize a lot. This is liberating a lot of nitrogen and phosphorus in sort of across that spectrum of decomposition, and you have available nitrogen and phosphorus to both microbes and crops, because it's almost like thinking you always have a piece of organic matter in that 20 to 1 to 10 to 1 area in the decomposition scale. I hope that that made sense. I spent a lot of time trying to figure out how to explain it, and we'll see. I'd love feedback on that, and I can tweak my my explanation. So really moving on, the question is, how does the Haney test and soil health calculation fit into this, now that we know a little bit about how microbes relate to nutrient turnover? And so if you've ever seen a Haney test printout, you'll know that it gives a lot of different numbers. But really the key numbers in terms of thinking about the soil health calculation and thinking microbial activity and nutrient turnover is this water extractable organic nitrogen and water extractable organic carbon. Excuse me, I have WEON twice when it should be carbon and nitrogen. And then these, it also measures inorganic nitrogen and phosphates. It measures that organic matter content. And then there's that Solvita test, which is that the amount of carbon dioxide that's respired by the microbes in a 24-hour period. And so this is pretty small. This is one of Paul's printouts of his results, and just to sort of orient you with where you'd find these numbers on a Haney test, and how these numbers are used, or these are the numbers that are used for the soil health calculation. And it's really, at the heart of the soil health calculation and thinking about microbial carbon is that feed value, that water extractable organic carbon ratio to water extractable organic nitrogen ratio. And Paul touched on this, is that Dr. Haney thought it was really important to use these water extractable numbers instead of the soil organic carbon and soil organic nitrogen, because it's these liquid sugars and nitrogen, and these liquid sugars, the liquid carbon and nitrogen that are really driving microbes at any given second. They're the liquid carbon and nitrogen that's giving microbes the energy to complete the rest of the decomposition. So the soil health calculation, I don't know if any of you have actually looked at how it's described, especially on Ward Lab, but it looks like this, which is just sort of right? Like, I don't know, it doesn't seem that exciting or interpretable. But really, when you think about it in terms of what we've been talking about, is it's that Solvita, that CO2 respiration number, so microbial activity, over microbial hunger, and which is just an inverse of that feed value. So the hungrier the microbes are, the higher that carbon to nitrogen ratio is. And then you add on this they call it, they sort of credit the soil. If there's a lot of water extractable organic carbon, they're going to credit the soil some points for soil health, because it means there's just more fuel for the microbes to stay active, to start to continue to make enzymes, to continue to decompose. And then they also give credit to water extractable organic nitrogen, because that's really the key to how much of that organic matter is in that sweet spot for plants, how much of it is being decomposed in a way that microbes can share with the plant. So in a way, the soil health calculation, the way Dr. Haney set it up is really it can be interpreted as a willingness of microbes to share nutrients based on this water extractable feed value. And so here's sort of shows you how soil health calculations can change depending on how your activity and water extractable values change in your soil. So it just these are hypothetical numbers here, but when we have less active communities and hungry microbes, this Solvita over WCON WO to WEON is going to be low, and when you have more active and satiated microbes, then you're going to have a larger number of like 15 I have here as an example. But then when you add that, so here I have, where's the pointer? Sorry. So I've taken the 2, and then I've moved the 2 over here, etc. Taking the 15 and moved the 15 over here. And then when we have like a low fuel environment, so let's say around 50 with the water extractable organic carbon, we're going to divide that by 100 plus not a lot of nitrogen available, so around 10 divided by 10, you'll get a soil health calculation of 3.5, which is very low. Whereas when you have higher fuel but still low nitrogen, like in this case, in this cell here, then your soil health calculation starts to go up. It's above that threshold of 10 that they like to see, that Ward Labs likes to see. But there again, when you have a lot of nitrogen but not a lot of fuel to keep microbes active, you're also going to have not a great soil health number. So it's really when you have all these pieces together of an active community plus this a good carbon to nitrogen ratio plus extra fuel for microbes and extra nitrogen for crops, that's when you're really going to see higher soil health calculation. So here are some of Paul's results, and I've just summarized them so we can all try to wrap our heads around them instead of looking at the Ward Lab printouts. But these are Paul's done a lot of sampling, but and correct me if I'm incorrect here, Paul. Here we have one comparison that Paul did. These two here. Oops, excuse me. These two fields here, they were sampled in the fall of 2014. One of them is a long-term permanent pasture, and you can see it has a nice high organic matter content of 5.5. And then contrasted with a pasture that had been cropped, but cover cropped for the last 5 years with some winter grazing, as Paul described. And, you know, what I'd say here is that both soil health calculations are good. The long-term permanent pasture seems a bit low for a long-term permanent pasture, but what I noticed when I actually looked at the numbers is both of these sites had high activity, so high Solvita tests. They had the water extractable organic carbon, that fuel available, but they both had sort of a low feed value, so higher carbon to nitrogen ratios. And I'm not sure. So I'm not sure if that has to do with time of sampling, and we can maybe talk about this a bit later. But I want to move through the results. But just to give you guys an idea of what I'm looking at when I see a Haney test. And then here's another comparison Paul did in this just this last summer. So he took samples from his permanent pasture that's grazed and then a neighbor's permanent pasture that's grazed a little differently. And you can see these again both have nice soil health scores. I don't know if Paul was a little disappointed to see his neighbors is a bit higher than his, but both are very good. And you know, nice organic matter content too. And what these show is that they had high everything, and that's why their soil health, their soil health numbers are high. It's because there was high activity, a lot of fuel, extra nitrogen, and a good feed value for the microbes. So here's another set of results from Paul's sampling. Oh, I don't know what I'm doing. Here we go. I'll just run you. So Paul sampled 3 different pastures, one that with no cover crops, one that has cover crops with grazing and with winter grazing, and one cover crops with no grazing. And he sampled these in the summer of 2014 and then again in the spring 2015. So a lot of sampling at these sites. And what you can see is it's sort of the exact opposite of what we'd expect, is that the soil health numbers for the no cover crop field in both summer 2014 and spring 2015 were higher than the cover crop fields. And in the case of summer 2014, I'm not sure if the drought in Iowa had something to do with the fact that these are lower than in 15. But we see that there was, when I looked at the data, there's actually, the no cover crop field just had a really active microbial community, whereas the cover crop fields had low activity. And this is something I'm going to touch on, or I've briefly mentioned, but I think is really important, is the one drawback to the Haney test, and I don't really know how other people address it, and I think this is an interesting point of conversation, is that all these measurements involved in the Haney test, activity, water extractable organic carbon, water extractable organic nitrogen, inorganic nitrogen, they're all very time sensitive. So they really depend on when you're sampling. Are your plants sort of at peak biomass? Are the roots really active because it's close to flowering? Did you just harvest and you haven't planted your cover crops? So although this field has had cover crops for 5-10 years, does it not have any roots in the ground when you actually sample? Things like that. So that's why repeated sampling, but also replicates within the field, and then mirroring your time of sampling are really important. But what I'm going to do is get back to these results, get back to Paul's results when we have the phospholipid data, after I've explained a little bit about the phospholipid test and what it can tell us, and that gives us a bit of a fuller picture of what's happening on the Ackley farm. So I'm just going to move forward, tempted you with some cool data, but we're going to just continue on and talking a little bit more about this diagram and these relationships. And you might notice that I've added right here the term resilience, and this has to get to the point of, you might measure something using a Haney test, but are those results consistent? If you measured again in 2 months, are they resilient? Are you still going to have high activity? Are you still going to have a high soil health number? And the Haney test can't really tell you, but that's where I think can't really tell you this, but that's where I think the phospholipid fatty acid test really comes into play, is because it tells you something about the microbial community. And there's a lot of research to support this, that that specific metrics of the microbial community can start to tell you something about the resilience of soil health. And that's because it's the microbes who are actually living in the soil doing these transformations. So if you have a healthy community, a community that's structured in a resilient way, then the health that they're creating is also resilient. And so what exactly does this look like? So the phospholipid fatty acid test is by no means the only way to measure microbes, but I think it's a good one, because and there's a lot of disagreement on this, but I think it's a good test because it tells you, it can measure really well the abundance of fungi and bacteria and their relative abundance. And it can measure pretty well the abundance of gram-positive and gram-negative bacteria. And what it does is it just measures the fatty acids in the cell wall of microbes. So it takes a soil sample, it kills all the microbes in that soil sample. It extracts uses awful organic solvents to extract the fatty acids in the cell walls of those microbes, and then runs that through an analyzer, and you get a profile of different fatty acids. And it does do this. It does measure fungi and bacteria and gram-positive and gram-negatives in a meaningful way. And so the reason why I care about fungi, and especially gram-positive bacteria, is because these are your slow-growing microbes, and these are your microbes that are really what in the research over the past few years has shown us that are really contributing to resilience. You want microbes that are slow growing so that they're holding on to their nitrogen, which may not sound good, but overall as a community, you have a large community that's just turning over slowly. So it's sort of like always giving something to the plants, but not a boom or bust situation. And that's why I look for fungal to bacterial ratios and gram-positive to gram-negative ratios. I just want to give a quick comparison of, I won't go over this too much, but for those of you who are interested, how the phospholipid test compares to other methods. There's the Ingham microscope method. It's really easy, it's pretty cheap, but it's really subjective and it misses a lot of information, and I don't think it's very accurate from the experience that I've had. Microbial biomass is a decent measurement, but it's not really related to activity. So you can have a big population, a big community of microbes, and they might not be very active, and it's really the activity that's going to tell us about nutrient turnover. The phospholipid test is, it's starting to get a little bit more expensive, and the bills add up for sure, but it is very good at what it does. And then you can get into very expensive analyses that labs are just starting to do, and this is what the researchers in the university are doing, which is looking at DNA and diversity. And it gives you a very detailed analysis of the microbes there, but it's not really sure how we relate that to function yet, especially in a very practical sense for a land manager. So I don't think it's quite the measure. It's not quite there in terms of farmers using it to tell us anything to do with fertilizer recommendations, for example. And then just to compare that to the Haney test, which is, as we know, measuring microbial activity. So here's an example of just it happens to be, I don't work for Ward Labs, I have no affiliation with them, but they do happen to be the ones doing the Haney and phospholipid test in a reliable and affordable way. So here are some of Paul's results, phospholipid results, and I'll just walk you through what the report looks like. And you may, what I'm about to say might be slightly disappointing. You see a lot of different measurements on this test, but I really think only a few are valuable. So if you were to do these tests, I would suggest you really focus on this fungal to bacterial ratio, the FB ratio, because it's this proven indicator of resilience and resistance of microbial communities. We know that microbial communities with higher fungi to bacteria store more nitrogen, have better water holding capacity, and just contribute more to soil structure. So it's a really good indicator. And the test actually comes with a rating system that's really fairly decent. So you can say when your fungal to bacterial ratio is greater than 0.35, it's excellent, etc. And then the gram-positives are often slower growing bacteria. And so when you have a higher gram-positive to gram-negative ratio, you can say not with as much confidence, but you can say that the community is more resistant and a more resilient community. And this is resistance and really resilience to things like drought. How well will this community withstand things like climate change and drought and the impacts of just general soil disturbance. Then some other interesting things the phospholipid test gives you is it gives you this estimation of the size of the community, and I like it as a secondary indicator, just as a comparison to see if everything is making sense. But as I said before, biomass does not mean activity, so it's not incredibly useful. And then another thing that I like to look at, because I am a microbial ecologist and specific organisms are important, is the fact that the phospholipid test also gives you an idea of how many actinomycetes, so these fungus-like bacteria you have, how many N symbionts you have, how many are the biomass of our buscular mycorrhizal fungi, and then the biomass of the saprotrophic fungi. So the things not to focus on is this predator-prey relationship. Phospholipid fatty acids doesn't do a very good job at measuring predators. So it's really dependent on time. So I would just ignore this section. I would also just ignore the section on all the specific fatty acids and what they might mean, Not to say that it's not interesting, but there's not really any solid scientific links to soil science, links to soil health and resilience and resistance. So I feel like this test with the fungal to bacterial ratio and your gram positive to gram negative ratio, you sort of have enough going on, and it's very valuable information. I don't know. I don't think we have time to go over this. This was just me explaining that Ward Lab uses around 4,000 of their samples in a histogram, and that's how they come up with their rating systems that you see on the phospholipid test. So back to Paul's test, incorporating now his soil health and his fungal to bacterial ratios and his gram positive to gram negative ratios. So here again we have the first set of results, which were these long-term permanent pasture and then a crop pasture with winter grazing, where soil health numbers were okay, but this long-term pasture, it seems sort of a bit suspicious in terms of, like, I thought it should be higher, and noted that there was this low feed value, this high carbon to nitrogen ratio. And looking at the phospholipid test, what I notice is that the fungal to bacterial ratio is very low. It's very poor. There are more gram positives, but it sort of is an alarm bell to me that it's dominated more by bacteria. And so I don't know if Paul can think back on the long-term pasture and think about, anything that might be there that might not be promoting fungi, if there's a huge mat of, old biomass or something. But maybe I'll get him to speak on these comparisons, and then we'll move on to the next set. Just to note too, this the comparison of Paul's neighbors to Paul's permanent pasture. You can see here there's nice balanced communities, and that there's average or slightly above average fungal to bacterial ratios. So they aren't like off the charts, but the microbes that are there are cycling a lot. They're active, and they have reached sort of a nice equilibrium with the soil. So I will stop here and see if Paul has anything to add or can think of anything that might be not making the fungi happy in that long-term permanent pasture.
Sarah, I'll start with the neighbors thing at first, and that was kind of a joke between him and I. And I was really surprised because we'd been doing rotational grazing, about 50,000 pounds to the acre on that for 4 or 5 years, and it had been seeded for almost 40. But his ground had been seeded since about 1960, and Lance thought probably that extra 15 years was what made a lot of the difference. Neither one of those permanent pastures probably had very much erosion on them at any time. Probably about as good a soil as we could get on that type of a location. The other two, the two at the top, I'm a little bit at a loss of just what went on there. Lance tried to explain it to me, and I think the permanent pasture, I think I got into, probably I just grabbed one sample for each one of them. I didn't take 3 or 4. I think I probably got into something that wasn't representative of maybe the whole field. But that's about all I can say about it.
Yeah, well, that's really great information, and I think I'm going to talk a little bit about replication and what can happen with a single soil core. So I'm really glad you brought that up, Paul, because yeah, I think that's a good point. When it's a hard test, it's expensive, and sampling takes time, but then it's hard to interpret sometimes. But I think it at least this gives us a good idea of sort of a baseline of what's happening in these pastures. Okay, so let's look at the other set of results. So this was this nice comparison that Paul has with no cover crop fields, and then cover crop with grazing, and cover crop with no grazing, where he sampled in the summer of 2014 and again in the spring of 2015. And you can see here that, again, this no cover crop field is slightly above average and is just sort of performing a little bit a little bit better than even in the microbial sense than the other two fields. There are more gram positives, and then just some weird strange inconsistency. So the fungal to bacterial ratio in this cover crop field without grazing is very poor, but there are lots of gram-positive bacteria. So I actually was really, and I still am, a bit stumped by these results. And so I'll let Paul chime in and see what he has to say. But part of me thinks it might time of sampling, maybe to do with the drought in 14, and maybe to do with the fact that it was a spring versus summer sampling. I'm curious to see if Lance had anything to say. Or maybe it's just an issue of, again, like not a good representation. It is really hard to get a good representation of microbes in the field. You do need to take replicate soil cores as well as replicates in your field. But I'm gonna turn over.
Okay, thank you. Yeah, that's a puzzle, and Lance didn't go into that, or I didn't visit with him about it. The first sample is definitely the best. Those are all 1 acre plots that we measured out for this. It was part of a SARE grant. That's how I was able to afford all those soil tests. But anyway, it was dry when we took it all. It was right after wheat harvest, the one that was taken this summer in 2014. When we planted the cover crop, the other 2 samples, it began to rain last summer about in August, and the cover crop, the forage sorghum, literally about drowned out in these samples here. I just was really surprised, and I didn't realize there was that much different in those soils in that short a time. So I will say that that top sample definitely has the best soil probably of the whole set of samples, probably the best drained, definitely the best position when we got into something where we got a lot of wet weather, which actually stunted our forage sorghum a year ago in the fall of 2014. That's about all I can add to it. We took them this spring just before we planted the others, and the one that was grazed we turned about 130,000 pounds in there, cows and calves. They stayed 6 hours, and they had it about 90% flattened when they left. They didn't probably eat more than half of it. So there was a lot of forage sorghum in it, not many nitrogen fixers. That's about all I can add to.
That's really helpful. I was wondering that, Paul. I was wondering, is that no cover crop field just sort of a better field? Because it certainly looks better, and it's consistently better, and so that makes sense. And that's the really tricky part about soil sampling, and designing how and when you take your soil samples. Dick Thompson, one of PFI's founding members and the great proponent of on-farm research, talked about that, of not having these randomized replicated blocks and then realizing that it was soil type that was driving all of his results. And so, but sometimes you just can't randomize and replicate. So, but what this, yeah, maybe we can touch back on these results during the overall discussion. I just want to get to a few more slides. I did actually have this randomized replicate statement, just because I think it would be an interesting point of discussion. And I know a lot of people, there's even a question in the chat box of, how do we sample to really, we're spending all this money on soil tests, how do we sample so we can get the results we need? And I don't have all the answers, but I've certainly liked the conversation. And this is something that, so Paul touched on this, Lance at Ward Labs, he said maybe Paul's one soil core just was really funky. It just had no fungi in it or something like that. And this is a great example of that. And why interpreting soil tests is so difficult, which is maybe not the best way to end this session, but I love this diagram. It's from Tim Parkin at the Ames USDA lab, and from his 1987 paper. And so he took one soil core. Here you can see here it's 98 grams total, And he started to divide it up. He divided it first into 3 cores. And he found when he measured activity that the activity in the first core was way higher than the activity in their 2 cores. So then he went and divided that top core, top segment into 5 segments. And he found that this one segment up here was contributing almost all the activity. So he wanted to find out what was in that little segment. And what he found was a tiny little piece of organic matter that weighed 0.8 grams, and it was contributing essentially all of the activity that he was measuring. And so what had happened if that little piece of organic matter was not sampled? You know, it goes to show you that the scale at which microbes are operating and this nutrient turnover is happening is really an important thing to focus on and to consider. And so that's why taking replicate cores is so important, and that's also, and then why taking replicates within your field and sending, 3 separate samples of 10 cores is important too, because you're going to have different soil types in the same field. That's all I have for now. I'd like to turn it over to the group for more discussions, and thanks everyone for listening to my presentation.
Thank you, Sarah, for really taking such a complex topic and making it practical and understandable. And Paul, thanks for being the example of all of this and explaining how this works with you. So if anybody has questions for either of these two, please think about them, enter them in the chat box here, and we can start taking them as we go. And there's already looks like a couple of questions that have came up here. Carl was asking if in 3 to 4 annual rotation of produce crops that he uses winter cover crops with, do you know about how many Haney tests he would want to use to have a definitive statement of soil health and resilience? Any idea, Sarah, or how to hear that?
Into my microphone and I couldn't hear you for the last minute. Can you repeat that?
Yeah, sorry, Sarah. I just really repeated the question from Carl there in the chat box, if you can see that, about if he needs to do multiple Haney tests to get a definitive statement for soil health.
Yeah, I think I'd have to know a little bit more about what was going on, but I like tests midsummer when the plants are really active, or sort of not midsummer, I shouldn't say that, I'm used to annual crops, but when your crops are most active. So when they're going to be having the most interactions with the microbes, I think that's a really good time to take Haney test and a phospholipid test. I really do think that the Haney plus the phospholipid tells you something, that fungal to bacterial ratio is really important. You might want to take Haney tests, and then if they're not sort of giving you all the answers you want, then next year take a phospholipid test if you don't want to commit the money to all that testing. I totally understand that. But so I would definitely take the Haney test when plants and microbes are interacting most. Another thing to consider, I know a farmer here close to me he's asking the question. So he's an organic vegetable farmer, and he tills a lot. He's an organic vegetable farmer, but he also he has this really intense regiment of cover crops. 3-4 cover crops a summer, and he's really wondering is all this time cover cropping worth anything? Is he really even doing anything for his soil health? And so we devised a sampling schedule where he's sampling. So he also has a year where this field is just cover cropped, and then a year when it goes into production but is also cover cropped. So he's sampling the soil after it comes out of a cover crop year, and then he's going to sample at the same time the next year after that same soil comes out of a production year, and to see if there are any changes. Is that sort of benefit of cover crops maintained over the production year? So I don't know if I'm at all helping answer Carl's question. I'd probably have to, talk to Carl a bit more specifically, but I would focus on when plants and microbes are interacting.
Yeah, great. Thank you, Sarah. And Paul, it looks like Wendy has a question for you. Wendy's asking here, what's your plan next? What are you gonna? We were looking at your results, I think, when she asked this question. So she's wondering if you're gonna utilize those and make any changes or take more tests, or what's your plan?
Boy, that's a tough question. Looking at those results, you just about really wonder what you're doing. But from what I've seen with what in the field when we harvest something, we're going the right way as far as reducing our fertilizer. I've definitely went too quickly with the nitrogen. When I talk to some fellows that are further ahead of this than I am or further along with it, they're starting to talk about 90 pounds of N for 190 bushel corn. And so I know there's things ahead of us, but you really get some of those puzzlers. I think one thing we'd change if we do any more test strips, it's going to go across the whole field. It's not going to be. I thought those 3 acre pieces that we laid out were really closer in ability than they were, but when we got excess rain, some of them didn't have the drainage that they needed to produce a really good crop. The first one probably did, and it did have a lot of volunteer wheat on it. I mean, it was just thick with volunteer wheat, and I didn't spray it out, that one with no cover crop. So in a way it had volunteer, it did have a cover, but wheat is not very friendly to microbes or mycorrhizae, as I understand it. Uh, I hope that helps. In a perennial system of rotational grazing, I'm going to jump in ahead, I guess, of Sarah for a minute. I don't know when's the time to take those samples. I'm going to say when you want to know something, and in the CRP I would definitely take samples before you start grazing it, if that's possible, and then take some afterwards. So I'm going to close with that for now.
Yeah, thank you, Paul. Sarah, if you don't mind, is Wendy's question about adding topical fungicides if that kills the fungi below ground, and then also if you could also address Kayla and Landon's question next about the best time for taking the test for rotational grazing in the perennial system.
Yeah. So Wendy, I have not read the literature on effects of topical fungicides on soil, but I would guess that those fungicides are still on the residues that are, and so there is going to be a residual effect in the soil, or could be. It might be something you'd want to test. I'm not sure if research has been done on it. It's a really good question. Fungi are pretty sensitive. They're very sensitive, and so I'd be hesitant to say that topical fungicides aren't affecting fungal networks below ground. Now let me read. I'm a slow reader, so I'm going to read Kayla and Landon's question. Yeah, perennial rotational grazing systems. Yeah, I think late spring would be a good time to test, or just at summer solstice. So mid-June, mid to late June. That seems to be, yeah, I would say I think you're right, Kayla and Landon. That's at least a good start, and it's probably where I would start when I would sample. And then we're also, yeah, so I would just sample, since you want to compare your CRP to grazing, I would sample your CRP at the same time you're going to sample your grazing, so that at the very least your time of sampling is comparable. Does that make sense? So if you're going to be sampling your rotational grazing moving forward in that late spring window, then sample your CRP, your baseline CRP in that same window too, so that at least you can't say, oh, well, the differences in the results are because they were sampled at different times. And I think that that's a fine time, in general, to sample grassland. So there's no criticism there. And then Eric's question. Oh, yes.
Go ahead. Go ahead, Sarah.
You go ahead, Paul.
Okay, I would suggest one thing other to Kayla and Landon to, if you can mob graze that or high density graze it in small paddocks, it'll increase your nutrient cycling by a bunch. It'll do it probably as fast as anything you can do to it in that CRP. That's all.
Right, and the reason for that is really stimulating those roots and that root turnover, and so that's thinking about getting that liquid carbon and nitrogen to the microbes and getting lots of fresh inputs of dead roots as the roots cycle, and so that's exactly why mob grazing and rotational grazing are so effective at really stimulating and feeding those microbes. That's a great point, Paul. So to Eric's question. Yeah, so wood chips. Wood chips are great for white rot basidiomycetes. So those are the wood degrading fungi. And so you're going to, over time increase your fungal biomass with wood chips. But they're also really, really high in carbon. So they have that very low feed value. And so I think in an actual production setting, wood chips on a large scale aren't a great idea. The reason why cover crops, so I actually haven't done a lot of reading on this, so don't, completely quote me. But the reason why cover crops are so great is because they're in that nice range of feed values for the microbes. They're a little bit higher, maybe 40 to 1. And so the microbes can get it down to the sweet spot for themselves in plants pretty quickly. And so you have sort of faster decomposition. And so when you have faster decomposition, you have more incorporation of organic matter into the soil, and you're making those improvements faster.
Just thinking, well, how much wheat straw or rye straw can tie up nitrogen or what
Exactly.
or food in the soil. Wood chips could do an even better job, and I think it'd be nice to do a little experiment maybe before trying it on a field scale project.
Yeah,
would be my thought.
Exactly. Yep.
Yep. Well, I wonder if anybody. Okay, here's Laura with another question. Maybe we can take that real quick then.
I've been struggling with that. I don't know what I'm going to do, whether we're going to stick with regular soil sampling with the Haney. I think if I go by what Dr. Ray Ward was showing us, you could probably guesstimate where you'd be with a Haney test close enough if you wanted to start, if you'd done a few here and there. When he was showing how closely the two paralleled each other, that kind of answered that, I think. That's about all I can think of on that, Laura. Thank you.
Yeah, so like as Paul said, you get that water extractable carbon to nitrogen ratio out of your Haney test, but research has shown that it correlates very nicely to your organic carbon and nitrogen ratios. And so I'm a microbial ecologist. I'm pretty biased in answering this. I think microbial activity is a really the Haney test is a great test, but it's just the time of sampling has to be taken into consideration. One thing that I do really like is I do really like the carbon to nitrogen the straight up carbon to nitrogen ratio, because although those values are correlated, it's nice to just know exactly what your carbon to nitrogen ratio is to start out with. So that doesn't really answer your question. I think they're both useful, maybe at different times for different reasons.
The next question on what would the cost of a Haney soil. I'm not sure what just the cost of a Haney is. I'm thinking the Haney plus the PLFA was either $39 or $49 a sample.
Yeah, I don't know. Maybe Stefan knows. Haney is 49. I think it's double that for a phospholipid test, approximately. It's expensive. It starts to add up. So Stefan's question is, given the availability of soil test results, do you think the variability, do you think they're currently actionable enough for farmers? I think it's so important to conduct replicates of these soil tests because microbial activity is so variable. And so, for example, in a perfect world, in that example of Paul's where he has the no cover crop and the cover crop with grazing and the cover crop with no grazing, which is such a beautiful experimental design, it would be great, as Paul said, to sample across the field, or to set up that experiment across the field so that you have all those treatments across different soils, and then you'd sample in the different soil types and send all of those samples in. So all of a sudden, you have 3 times the number of samples Paul sent in. But I think doing randomized, if you have the money to do randomized replicated experiments, then Haney tests are great and they are actionable. But it's really expensive. So that's when something like the Cooperators Program or other research programs and other research grants, I think, need to get on board with helping farmers pay for randomized replicated testing of these really important tests. David A., are there fungi which are detrimental to soil health? Yeah, there are pathogenic fungi, but by and large, the fungi in the soil, those saprotrophic fungi, are beneficial. And then I'm really not that familiar with fungicides, but there are not species-specific fungicides as far as I know. So some, topical fungicides are targeting those pathogens, which might only be affecting the crops and not necessarily the soil. But as I said, fungi are very sensitive. So they're hard to study because they have such large body masses with all of their mycelium and networks in the soil. So it's hard to know what an individual is. So it's hard to know what a species is. They're a tricky organism to understand well. But I would say, by and large, fungi should be considered beneficial to soil health.
I think I'll go ahead and say real quick, while there's a lull, if somebody else has a question, feel free to type it in. We got a couple minutes left. But we did actually host a Farminar last December where we had one of our PFI farmers, Fred Abels, come on and do a presentation along with Lance Gunderson from Ward Labs, who has been referenced quite a bit tonight. And they talked about the Haney soil test and the PLFA test, and so that information could be really complementary to what we're talking about tonight. So if anybody wants to look that up, I'm going to paste the link here in the chat box. You can watch that archived recording. And again, you'll be able to watch this archived recording here by the end of the week.
I guess I would make one comment on Laura. I assume she is asking about the pH test. What I really liked about taking soil samples over a period of time was that last slide. It was over a period of time our pH was creeping up or remaining the same, and I felt like we were getting the nutrients were becoming more available, and I think that was one of the biggest things that I got from those soil samples that were taken over a period of time. The cation exchange capacity was going up, and the organic matter was very creeping up, and the soils were becoming better just because of the way they were being farmed. But I think the pH tests are valuable over a period of time, or when you're starting with a piece of ground.
Okay, well, it looks like activity in the chat box has kind of slowed down, and we've kind of hit our 90-minute spot here. So I'm going to go ahead and say that we're going to wrap this up and say big thank you to Paul and big thank you to Sarah for putting a lot of time into these presentations and really making this, kind of a complex topic understandable. So thank you to both of you so much for all the time that you put into this. Appreciate it. And thanks everybody for tuning in.
Thank you, Steve and Sarah. I appreciate it too.
Everyone for listening.